Hello students. Welcome to Land of Geeks. My name is Abdullah Arsalan. I have 8 years of research experience in biotechnology. Thank you for connecting with us. Keep watching, stay connected. Hope you are doing good. Let's start our today's session. Here are some topics that we are going to cover in this video. Through this video, I am going to explain the de novo pathway for purine biosynthesis in details, including illustration of de novo synthesis, location of de novo pathway, biomolecular involvement in purine biosynthesis, key steps in de novo pathway, whole set of reactions involved in de novo pathway, regulation of de novo pathway. Let's explain de novo synthesis. De novo is a Latin phrase, literally translating to from the new, but implying anew, from scratch, or from the beginning. De novo synthesis refers to the synthesis of complex molecules from simple molecules such as sugars or amino acids, as opposed to recycling after partial degradation. For example, nucleotides are not needed in the diet as they can be constructed from small precursor molecules such as formate and aspartate. The de novo synthesis of purine nucleotide means using phosphoribose, amino acids, 1 carbon units and CO2 as raw materials to synthesize purine nucleotide from the beginning. It is the main synthesis pathway of nucleotides. Let's discuss about the location of purine biosynthesis through de novo pathway. Location of purine biosynthesis. Purine synthesis occurs in all tissues. The major site of purine synthesis is in the liver and, to a limited extent, in the brain. Let's talk about the biomolecules involved in purine biosynthesis. Purine biosynthesis requires ribose 5-phosphate, glycine, glutamine, water, adenosine triphosphate, guanosine triphosphate, carbon dioxide, aspartate as substrates, and guanosine monophosphate, adenosine monophosphate, glutamate, fumarate and water are the end products. Now we will discuss the important steps involved in the purine biosynthesis through de novo pathway. Key steps involved in the biosynthesis of purines. Step number 1, ribose 5-phosphate, as provided by the pentose phosphate pathway, is converted into PRPP, phosphoribosyl pyrophosphate, by PRPP synthetase, in a step requiring 1 ADP. Step number 2, in the committed step in the process, an alpha amino group is then added to PRPP from glutamine to form 5-phosphoribosylamine. This reaction is catalyzed by glutamine PRPP imidinotransferase. Step number 3, a series of 9 reactions results in the formation of IMP, inosine 5 monophosphate. Step number 4, IMP can then be transformed either to GMP by IMP dehydrogenase, or to AMP by adenylosuccinate synthetase. Now we will discuss reaction of de novo pathway in details. The purine base is synthesized on the ribose moiety. 5-phosphoribosyl 1-pyrophosphate, PRPP, which provides the ribose moiety, reacts with glutamine to form phosphoribosylamine. This first step in purine biosynthesis produces N9 of the purine ring, and is inhibited by AMP and GMP. The entire glycine molecule is added to the growing purine precursor. Then C8 is added by 10-formyl tethahydrofolate. N3 by glutamine, C6 bicarbon dioxide, and 1 biaspartate, and C2 by 10-formyl tetrahydrofolate. IMP, which contains the base hypoxanthine, is generated. IMP is cleaved in the liver. Its free base, or nucleoside, travels to various tissues where it is reconverted to the nucleotide. IMP is the precursor of both AMP and GMP. IMP reacts with aspartate with the expense of GTP to form adenylosuccinate, which after release fumarate to form AMP. IMP undergoes oxidation to form XMP, xanthosine monophosphate, with the help of NAD, nicotinamide adenine dinucleotide. XMP later utilize glutamine with the expense of ATP to form GMP. The reduction of the ribose moiety to deoxyribose occurs at the diphosphate level and is catalyzed by ribonucleotide reductase, which requires the protein thioredoxin. After the diphosphates are phosphorylated, DATP and DGTP can be used for DNA synthesis. Purine bases can be salvaged and converted between free bases, nucleotides, and nucleosides by a series of reactions. Let's talk about how de novo pathway is regulated. De novo pathway is regulated by various allosteric enzymes through the process of feedback inhibition. PRPP synthetase is inhibited by the presence of AMP, IMP, and GMP. Glutamine PRPP imidinotransferase is inhibited by the presence of AMP, IMP, and GMP. IMP dehydrogenase is inhibited by the presence of GMP, and adenylosuccinate synthetase is inhibited by the presence of AMP. Inhibition of de novo pathway by pharmacological agents. Since tetrahydrofolate is involved in two reactions of de novo purine synthesis, folic acid analogues, such as methotrexate, inhibit the formation of tetrahydrofolate and thus interfere with purine synthesis. Thanks for watching this video. This was the detailed explanation of biosynthesis of purines by de novo pathway. We will discuss the salvage pathways for purine biosynthesis in details in our next video. For more articles on biotechnology, advanced sciences, engineering, mathematics, physics, and various other domains, please visit our website lambdageeks.com. If you find our content useful, please share, like and comment on our video, and do subscribe to our YouTube channel. Please visit our website lambdageeks.com for more articles on technology, engineering, mathematics, and various other domains. You can check our other website as well. We have features like YouTube Trends, Twitter Trends, Scientific Calculator, and many more other tools. If you like our video, please like, comment, and share. If you have any questions, please comment and we will resolve your query.